Thank you. I'm very honored to be here. And what I would like to talk to you about today is how genes can shape both investment and human capital formation in the long run. So the goal of the research is to understand the interplay between genes, investment, and human capital, and the way that genes can influence and shape the way that parents and everybody, each individual, invest in human capital, and how this has an effect over the life cycle. The background to this research is something that Professor Heckman mentioned before. There has been a lot of talking about unobserved endowments, about ability, about things that we cannot observe but that make people different. But in 2003, there was a great discovery, a great step forward in our understanding of genetics. The Human Genome Project mapped the DNA of every person, my DNA and your DNA. And so we have finally a blueprint of human development, of which part of the DNA code have an impact on later life outcomes. And this has helped immensely to understand the biological mechanisms that genes have on impacting our biology and the way that we are and the way that we evolve over time. So this has helped to open the black box of unobserved heterogeneity and to understand better how genes and environment interact together in order to produce human development. This is very general, so let me try to be a little bit more specific. I'm going to give as an example looking at body mass index, BMI, and one particular genotype. The FTO gene, which is a gene in the 16th chromosome that we all have, and there are some variations in this gene, and this variation have been associated to body mass index and changes in obesity rates. But in particular, we know that BMI has increased massively in the past 30 years, and yet the genetic pool of the population has stayed the same. So how can it be then that up to 50 or 60% of BMI is heritable, and still we observe these differences over the life cycle? One example can come from this gene. So looking at research from Rosenquist and co-authors, we can see the evolution of BMI over different ages for two types of people. Those who have the TT genotype on the left side, and those who have the A genotype, which has been associated to obesity. And in particular, we can see differences in the environment that have evolved over time. So we're going to plot separately the evolution of BMI for those who were born before 1942 and those who were born afterwards. Why do we do this? Because there has been a massive change in what people have called the obesogenic environment. Now, and in the past 20 to 30 years, high caloric food, dense caloric foods are available and are cheap for everybody to have. And at the same time, instead of doing physical work during our jobs, most of the time we stay seated in front of a computer or behind a desk. And so physical activity has become more of a leisure rather than something that we do during our work. And what they observe is that depending on the genotype, there's a very different increase in the BMI of people who have the risky allele and they were exposed to the obesogenic environment. So they were born after 1942. With respect to those who have the obesity-related allele, but there was no availability of cheap caloric foods. Another example can come from looking at children. In this particular case, the source is the ALSPAC dataset, so some British children who were born in the 1990s. And they were followed since birth. And we can see that in the first 5 years of life, so before age 5, there is no significant difference in the average BMI of these children according to their genotype. On the other side, after age 5, the average BMI of the children who have the A allele, the risky allele, tend to be higher than the ones of those who have the T allele. And this is consistent with the story that there are different investments that accumulate over the life cycle and developed into differences in BMI. So our contribution is to integrate genes into a model of human capital formation. So we take insight from human genetics and we try to integrate them into a simple model of human capital formation that has been a workhorse of economics. And the way that gene and environment can interact is going to be modeled in two particular ways. One is a change in the cost of investment. So different genotypes can change the cost of doing different types of investment. And the other one is that the genotype can change the biology of our body and change the productivity of different investments. And as I was saying, we're going to look at this in a life cycle profile. So I'm going to use these graphs to explain the model in a way that I hope is clear for everybody. Consider one particular type of investment, say caloric intake, how much we eat, and one particular outcome in human capital. In this case, it's going to be BMI. And we can trace the way that investment as an input, map into human capital as an output. So one of the features of the model is that the genotype of the children can shift the production possibility frontier and can change the way that inputs are converted into outputs. The second feature of the model is to consider a change in cost. So for this particular example, consider two potential investments, calories and sedentary minutes. And both of them, the combination of the two of them, can produce human capital and BMI. So the more I eat and the less I exercise, the higher is going to be my BMI. And if the genotype changes the cost of investment, this is going to have a shift in the budget set, so in available combination of input and outputs that the people have. And this is going to change the way that the optimal investment of children and how this is going to map into BMI. This is the math behind the model. I'm not going to dwell too much into it, but again, the idea is that we can consider a cost of investment and productivity in investment, and genotype can influence both of them. Some example to be more specific about why we model things in this way. In terms of productivity, I'm going to talk about a very influential paper by Caspi and co-author that came out in Science in 2002. What they see is that they consider child maltreatment as a negative investment in human capital, and as output they consider antisocial behavior. And what they observe is that one particular genotype changes the way that child maltreatment has an impact on antisocial behavior. In particular, for those children who have a low level of MAOA activity, there's a higher risk of antisocial behavior in conjunction with higher and severe childhood maltreatment. And this is an idea of, similar to what I was saying before, of differences in the production function of human capital. In the terms of cost, I'm going to talk about the ALDH2 gene. Is a genotype that is fairly common in populations of Asian ancestry, and it has been related to facial flushing and drowsiness and palpitations. So the way that these genes work is it metabolizes alcohol, and so the people who have this particular genotype, who have one particular genotype, when they drink too much, they have facial flushing, they become red, and it's much harder for them to drink too much. So they become drowsy, they have a lot of higher problems in drinking. And indeed, we observe that they have a lower alcohol intake. So the way that we model this is that this genotype changes the cost of drinking too much. This has an effect on the investment, how much we drink, and in the long term, this will have an effect on an outcome of interest like liver cirrhosis, for example. In this paper, we're going to focus on the FTO genotype, which I talked about before a little bit. And it was a genotype that was connected to body mass index through genome-wide association studies in 2007. And since then, a lot of studies have replicated the connection between this gene and body mass index, and also have tried to understand, using both animal models and human models, the way that this gene functions and the inner working of these genes. So how does this gene work? It seems to be related to appetite regulation. So for example, there's a study that shows that it's very much related to an appetite stimulant hormone, which is called ghrelin. And it's also related to neural responsiveness to food images. There was this very fascinating study where they brought people into the lab and they stick them into functional MRIs to map their brains. And they showed them pictures of high caloric foods, so basically a hamburger, or low caloric foods like salad. And what they saw is that people who have one particular genotype had a much higher brain activity in the regions of the brain that were connected to appetite and satiation. So the way that we model this is to consider FTO as an increase in the cost of dieting, one of the particular investments that we're going to consider. We're going to bring the model to the data using the AVEN longitudinal study of parents and children. It's a very rich data set that was collected by epidemiologists in Bristol. And it's a cohort of children born in 1991. And they brought these children into clinic visits in regular times. And this gave us access to very precise measurements of both height and weight, and therefore BMI, and also investments that we're interested in. So they gave the children a pedometer so that they could measure how often they were moving and how much exercise they were doing. And they also asked how much they ate in the past week. And this allowed us to have both measures of physical activity and diet. So this is Bristol, and the region near Bristol in the UK is where these children come from. Some summary statistic, just to have an idea of what the data that we have is. Body mass index has been associated, the A risky allele has been associated to higher body mass index, both for females and for males. And if we look at caloric intake, so how many kilocalories per day these children eat, we see that children with the risky allele tend to eat more calories than those that don't have the risky allele. Now we can estimate the relationship between these investments and human capital and health and BMI. So we consider BMI as a function of diet and exercise, the two investments of interest, and we control for family characteristics and the past stock of health, the past stock of BMI, in order to understand what is the mapping between these inputs and the output of interest. And we allow genes to index all of the parameters of this production function. And what we're interested in is to understand whether genes change the marginal productivity of investments. Therefore, whether this functional form, the way that inputs are converted into outputs, depends on the genotype of the children. And what this table shows is that we indeed observe that there are some significant differences between children with one genotype and those who don't. So if you look at the top row, this is a dummy for identifying the children that have the risky genotype. And we observe that without controlling for anything, we see that this genotype is connected to obesity. So we replicate what other people have found in the literature. More interestingly, if you look at column 3, we introduce food intake and sedentary minute, the two investments of interest, and we interact them with the genotype. And we see that there's a significant interaction between how much food children eat and the genotype of interest. Therefore, the children who have this risky genotype tend to convert the same amount of calorie into a higher level of BMI. The other effect that we're interested in is the cost effect. So we would expect that if it is indeed harder for children to refrain from eating too much, and if there's a higher cost of dieting, then we would observe a higher level of caloric intake for the children who have the risky gene. And indeed, we do find that both for males and females, both males and females, even controlling for a lot of observed characteristics and covariates, including their BMI level and the BMI of the mother and other family characteristics, we see that children with the risky genotype tend to eat more calories. But there is no significant difference in their level of sedentary activity. Using these insights, we construct a structural model that takes into account the dynamics, and so considers the fact that if indeed there is differences in the level of investments and the way that investments are are converted into outputs, then there could be accumulation of these differences over time, and this can lead to the observed differences in BMI that we see in both the children and in adults. And this also will allow us to calculate counterfactuals and to consider the effect of particular policies that can change certain outcomes. So again, I'm going to show this graph, which is a show of the data. Again, we will try to match this data by calibrating the parameters of the structural model. And I think we do a pretty good we managed to fit the data pretty well. We see that at the beginning, in the first years of life, both the children with the risky genotype and the T allele tend to have a similar level of BMI. But as investment change over the life cycle, we see that there is an increased difference in the average BMI between these two genotypes. And now we can consider different policies. For example, something that a lot of people have talked about is to put a food tax. So since calories are very cheap, a way of counterbalancing this effect is to consider a 50% increase of tax on calories. And what we observe is that if indeed we increase the prices of calories, the genetic differences in BMI tend to fade out and to fade away. And this is consistent with the results from the cohort born before 1942, when the price of calories was not so cheap, and indeed we didn't find a lot of difference in their level of BMI. Another thing that we can do is to consider different levels of income. And if we reduce the income that is available to people, we observe a similar thing. Again, we see that the genetic differences in BMI tend to fade away. And this is consistent with the results from Malawi. There are some researchers here at the University of Chicago who went to Malawi. They genotyped a lot of both children and adults, and they saw no connection between the FDO genotype and BMI, which makes sense because if we believe that the effect is not a direct effect of the gene but goes through investments, if you are in an environment where there's no abundance of caloric intake, where it's hard to where you have to do a lot of physical exercise, then we would not expect to see genetic differences in BMI. And indeed, this is what we find. And I think that both of these two counterfactuals speak to the idea of the interaction and the interplay between genes and between the budget constraints and the constraints that people face when choosing how to invest in their human capital. Finally, another thing that we can consider is a school eating policy. So a lot of schools have been talking about making better food available to their children and to allow the food that is available in the canteen to be more healthy. And we can model this by considering a 25% reduction of caloric intake only in the first 10 years of life. So when children are actually in school. And then we can see what is that effect in the long run by letting the model run for longer periods. And we do observe that there are still some differences in BMI due to the gene, but the interesting thing is, I believe, is that we can see long-term effects. So there's a lower level of BMI with respect to the baseline model, even if we focus only on the first 10 years of life. So early interventions can have an effect only in the long run, also in the long run. To conclude, I hope that I've shown you the importance of taking into account how genes and choices of investment interact with each other and build on in human capital formation. And what I would like to talk about is some work in progress and some potential extensions of this work. So in particular, right now, we just got access to the Framingham Heart Study, a data set that looks at adults in the US. And this allows us to have greater variation in age of birth and also in food prices that people were exposed to. And so having a bigger data set that looks at longer time period allows us to better understand what are the mechanisms that can shape the interaction between genes and investment. And one word on potential extensions. So far, I've been talking only about obesity and about BMI, but this model can be used and can be changed to talk about many other facets of human capital. So we know that there are some genetic variants that have been associated to cognitive ability, and others that have been associated to other non-cognitive skills or character skills. And so by considering the right genotype, the right investments, we can try to speak and understand more about not only obesity, but also other facets of human capital that we're interested in. And finally, one other potential extension that I think could be very interesting is to look at gene expression. So DNA is fixed. The DNA that I had when I was 1 year old is the same that I have now and that I will have when I'm 50. But genes work through gene expression. And so the way that a gene is expressed in our body changes over the life cycle. That has been a study in 2005 by Fraga and co-authors showed that identical twins have very different ways of gene expression. And this level of expression has been connected to different environmental effects, to different investments that people have seen. So there's a study by Professor Ekman and Gabriella Conti that shows how the level of gene expression of certain monkey depends on how they were reared during the first years of life. And so what we can do is to consider genes not only as a fixed endowment of the individuals, but also how they evolve over time and how there's a feedback loop between investments and genetic expression. And that's it. Thank you.